Hello, my name is Subrata Panja. I'm going to talk about sparQ DBS Library Prep Kit for rapid extraction of high quality genomic DNA from dried blood spots and library preparation for whole genome sequencing applications. Newborn screening, a public health system that identifies conditions that can affect a child's long-term health and survival. Typically, babies are screened within 24 hours of being born. 1 in 300 newborns have a condition detectable through newborn screening. Most babies with serious but treatable conditions caught by newborn screening grow up healthy with expected development. If we look at the history of newborn screening, it started around 1960s when Dr. Guthrie developed first blood test for PKU. Since then, different kinds of tests have been added for screening of newborns and being adopted by different states in the United States of America and globally. And most recently, the latest development in newborn screening happened when International Consortium of Newborn Screening established in 2022. Typically, newborn screening happens through three primary methods using blood spot screening or using pulse oximetry or doing hearing screening. For blood spot screening, typically we use a few drops of blood from baby's heel to determine if the baby might have one of many serious conditions. Using pulse oximetry, we try to determine certain heart conditions called critical congenital heart disease. And using hearing screening, we try to determine if a newborn is deaf or have hearing problems. Throughout this presentation, we are going to focus only on blood spot screening, where I mentioned before, we use a few drops of blood from the baby's heel to generate dried blood spots or DBS and uses different molecular tests like mass spectrometry, qPCR, and targeted sequencing to determine if any serious defect present in the newborns. Quantabio is well known in the newborn screening field. Our reagents have been widely adopted by state laboratories for research programs identifying the underlying genetic mutations involved with inherited diseases. Our newborn screening solutions improve ease of use. Those are automation friendly, provide greater sensitivity, and are scalable for a wide variety of sample sizes. Our workflow starts from dried blood spots and then extracting DNA and then amplifying using amplification reagents in a qPCR machine for detection. For example, we have extractor DBS for DNA extraction from dried blood spot samples, different qPCR reagents for amplifications, including our premium tough mix product lines. And finally, we have qPCR cycler for detection. Current newborn screening method has a serious limitation. It can interrogate only a finite number of genes associated with specific genetic disorders, but novel or unknown disorders will be undiagnosed. One way to address that problem is by extracting high quality genomic DNA from dry blood spots and then sequencing as a whole genome format and then doing data analysis to identify all the mutations or the mutations we are trying to identify. Overall, whole genome sequencing provides comprehensive, reliable, and fast genetic information regarding genetic disorders present in newborns. One of the challenges is that current extraction methods do not support a quick and cost-effective extraction of high-quality gDNA from DBS samples. That is why we developed a quick and effective extraction method from extracting gDNA from dry blood spots and combining that with our premium DNA library preparation kit with enzymatic fragmentation. And both modules together called sparQ DBS Library Prep Kit, and it will be used for whole genome sequencing for newborn screening or sequencing. The workflow of sparQ DBS Kit has been tested in our lab by generating DBS using certain or finite amount of blood, typically 60 or 75 microliter of blood spotted on Whatman 903 paper. And then we took three 3 millimeter punches from dry blood spots to extract DNA. The extraction method is simple, streamlined, and it takes only 40 minutes. The protocol starts by collecting three 3 millimeter punches in a tube. Then we add extraction reagent to the tube, incubate 50 degrees centigrade for 20 minutes with shaking at 1000 RPM in a thermomixer. After that, we collect the liquid in a new tube and it goes through a typical magnetic bead cleanup process, and finally we collect high quality genomic DNA in a new tube. To understand the quality and quantity of the extracted DNA, it goes through a QC check or QC method where we used tape station to look at the integrity of the genomic DNA or quality of the genomic DNA, and we used qubit to look at or quantifying the amount of extracted genomic DNA. Here is a representative tape station trace of 7 samples extracted from dry blood spots, and you can see that the genomic DNA extracted from 3 punches of each DBS sample generated very high quality genomic DNA with the bands higher than 50 kb, and the Dean score is higher than 8, indicating we were able to extract high molecular weight, high quality genomic DNA using this method. From 3 punches, when we extract the DNA and quantified using qubit, the average yield was 175 nanogram. And we consistently get high yield from different days, different dry blood spots, and different sample types. Here is another example where genomic DNA was extracted from different number of DBS punches, and you can see that if we need higher quantities of genomic DNA, we can use 6 or 8 punches. And with 8 punches, we can get as high as 500 nanogram genomic DNA. Nevertheless, I want to emphasize the point that with 3 punches, you can get an average of 175 nanogram gDNA, which is sufficient for PCR-free whole library preparation, whole genome library preparation. To prepare PCR-free whole genome libraries using sparQ DBS Kit, these are the following steps. First, starting with genomic DNA in a tube, then we add frag and polishing mix to the DNA, incubated 32 degree for 4 minutes and 65 degree for 30 minutes, which will fragment the DNA, end repair and A-tailing will happen in one reaction. Our goal here was to generate an insert size around 550 base pair or higher. And once the DNA is fragmented, end repair and A-tailing happen, then we will add full-length Y-shaped adapter for Illumina sequencing and ligation master mix and incubate 20 degrees for 15 minutes, and then it will go through a bit cleanup, and finally we will collect our library. Each prepared library went through the QC check, which includes looking at the quality of the library by running it in a tape station, and we quantified the libraries using both qubit and qPCR. And finally, we pulled the libraries and sequenced in NovaSeq X. One thing I want to mention that PCR-free libraries with full-length Y-shaped adapter does not migrate properly in tape station or any kind of fragment analyzer system because the ends of the Y-shaped adapters are not perfectly double-stranded, rather it's forked. So it does not migrate properly. So what we need to do is we need to use few cycles of PCR using P5 and P7 primers and then linearize the libraries, and then we can run in tape station to look at the size of the libraries. Here is an example where the library size distribution was between 700 to 750 base pair for all these 4 libraries I'm showing here. That means library size distribution was somewhere between 550 to 600 base pair. We also generated 3 other libraries with specific input amount. For L5, we prepared PCR-free libraries with 400 nanogram input DNA. For L6, we used 100 nanogram genomic DNA for PCR-free library preparation. And for L7, we used 100 nanogram DNA for library preparation and amplified for 3 cycles. And in all cases, library size distribution is between 700 to 750 base pair, showing consistency of the fragmentation chemistry. Here we are also showing a correlation between input DNA amount and library yield. So in this example, 4 libraries were prepared with an input amount between 140 nanogram to 185 nanogram. And you can see that with that range of input DNA, we got library yield between 28 nanomolar to 42 nanomolar, which is more than enough we need for sequencing. Now, as I said, we try to get the fragment size around 550 to 600 base pair. Now, if for other applications you want relatively shorter insert size or fragment size of the DNA, instead of 4 minutes of fragmentation, you can fragment it for 6 minutes or 8 minutes to get relatively smaller fragment size. So that indicates that the fragmentation chemistry is tunable. Once libraries were QC'd, pulled together, then we sequence the libraries in NovaSeq X using 2 by 150 pair end reads. Our target output was 140 GB per library with target coverage was 30x per library. Here is some QC parameters from uh, the libraries we sequenced in NovaSeq X. You can see that the duplication rate was between 7 to 8%, and that was independent of the input amount of the gDNA, or addition of 3 PCR cycles did not increase the duplication rate. And in all 3 cases, the percentage of map reads were above 95%. That indicates sparQ DBS Library Prep Kit generated high quality DNA libraries with minimum duplication artifacts and a high percentage of mapped reads. I mentioned before our target coverage was 30x across all the chromosomes, and that's what we got with 400 nanogram input DNA, 100 nanogram input DNA, or 100 nanogram input DNA with 3 PCR cycles. We got uniform, more than 30x uniform coverage across the genome. And this is essential for determining most germline mutations with high confidence. Finally, this is a summary of all the variant calls we got from the sequence data. So we looked at total variants, total SNPs, structural variants, duplications, and deletion rates in either 400 or 100 nanogram PCR-free libraries or 100 nanogram libraries amplified for 3 cycles. And you can see that the total number of variants did not differ based on input amount or if it was amplified or not. So this indicates that libraries prepared with sparQ DBS Library Prep Kit generated low mutational frequency, which indicates less error incorporated during the library preparation stage. So what we learned from this presentation is that sparQ DBS Library Prep Kit provides a streamlined solution for GDN extraction to PCR-free whole genome library preparation. Rapid gDNA isolation workflow requires 40 minutes, and the PCR-free library prep takes 90 minutes. The extraction method generated high molecular weight gDNA with molecular weight higher than 50 kb, and we got high genomic DNA yield from the extraction. And finally, high quality libraries generated reliable sequencing data. Overall, the sparQ DBS Library Prep Kit provides a rapid, cost-effective, and robust solution for whole genome library preparation from DBS samples that will benefit newborn screening across the globe. With that, I want to thank you for your attention. If you have any question, please reach out to me, or if your lab is interested in whole genome sequencing based newborn screening or newborn testing, please reach out to us for a sample. Thank you.